Dear colleagues and dear audience, it is my great honor, and also thanks a lot for the kind introduction. It is my great honor being selected and also being awarded for the Goetz Prize. So I'm pleased to share my work in the past 7 years with Professor Adriano Aguzzi. So the main focus of our study is neurodegenerative diseases. As you know, that this disease has been described have been more than 100 years, and they occurs in different types of diseases. And in the last 30 to 40 years, a lot of efforts has been put on the studies for the protein aggregation, the metabolism, and also the drug targeting. However, although enormous efforts have been put into this direction, we have not been very successful in this field. This implies our understanding of the disease is still incomplete. So when I joined the Professor Aguzzi's lab, I got some inspiration because of my previous research at the Institute of Neuroscience, Chinese Academy of Sciences. I was there focusing on aging. And in the aging field, the genetics has played a major role because the first longevity gene was identified by genetic screen. And I also dissect that longevity is different from healthy aging. And I identify a new pathway that really control healthy aging in C. elegans also. From there, I understand doing research, you have at least two approaches. One is hypothesis-driven, one is hypothesis-free. So for hypothesis-driven, you can speculate which genes or pathways could be implied in a phenotype. Then you can test it. However, you can't know more than you already know. So that is according to Karl Popper. Using genetic screens, you can not only identify the known links, you potentially can identify something that is completely new, then you expand the field. So there, I really start to appreciate the power of genetic screens. If you look at the common biological research models, including yeast to human, to do large-scale genetic screens, it is feasible for yeast, for worms, for flies, but it becomes challenging and even impossible from zebrafish until human being. So it's a very important obstacle that we need to break if we need to do disease, especially for human disease. And in the last 14 years, since the development of CRISPR technology, for gene perturbation has been dramatically changed. And you have a lot of tools you can really do gene-level manipulation, including gene knockout, gene activation, gene silencing, and all epigenetic silencing. And indeed, at the genome scale, for human genome libraries, there you can have really already some approach. This is a widely used approach. So the strategy is the guide RNAs are really put in a bug. That individual cells are transduced with a lentiviral vector with low titer to ensure one cell gets only one gene perturbed. And one guide RNA enters around 500 to 1,000 cells. There, if the gene affects cell growth or a drug resistance, the guide RNA abundance in the cell population will change. After sequencing of the cell population, you will read which gene affects the phenotype. So the readout is not is an indirect reflection by next generation sequencing. So as I mentioned, so this strategy is really good for cell autonomous phenotypes like cell survival, drug resistance, or if you have a reporter, you can do selectable phenotype by FACS or whatever method you can select the phenotype of interest. However, if you do, we are focusing on neurodegeneration. The phenotype is a bit complicated. So if the cell is affected, the phenotype you observed in the cell that is transmitted from the other cell, but the protein aggregates spread from cell to the other one. So the phenotype you observed in the cell, if it's arised from a nearby cell or the other stimuli, you will not be able to detect by sequencing. So then we need to a new, completely different approach. How to do that? I mean, you can put the lentiviral vector one by one into the microwave plate. And each well, you capture the cells that have a homogeneous precipitation. Any phenotype you observe is really caused by this gene precipitation. However, this tool is really demanding, but it is very difficult to generate a construct. And also, you can really do much more complicated and important phenotypes, for example, cell secretion, biochemical or morphological or cell-cell interaction assays. So that really needs us to build a new tool to do neurodegeneration and also many other biomedical research phenotypes that can't be addressed by the pooled method. Another thing is, I need to mention, in the pooled method, the single guide RNA they used the efficacy is difficult to predict, so we need to solve the issue for array library. And also the reference genome, the previous library were designed based on a reference genome. However, in the nowadays, everybody is different, and also there's a lot of scenario we need to use primary cell culture and iPSC lines, so we have to consider DNA polymorphisms. Then finally, we decide to generate 2 CRISPR array library for both gene loss of function and gain of function studies. Now the biggest challenge is how do we make it? So the first question I solved is, if the single guide is variable in efficiency, I try to titrate how many guides if we target to same gene that can increase a robust gene editing efficiency. You can see here in HEC293 cells, with putting 4 guides, the efficiency for gene activation is dramatically improved and is very robust. For gene knockout, if you don't test a single guide, you don't know the efficacy. With the 4 guides, they are very robust, and the result is confirmed by long-read sequencing of the edited cell genome. So then with the 4 guides, I really get encouraged that we should be able to make a new array library, and this library is very efficient. Now the next challenge is how can we build it? Even one library, even one gene, we have 1 plasmid, it will be more than 2,000 genes for the human genome. And even we want to make 2. So the traditional way of making molecular cloning, if some of you may know, the most important thing is you need to make single colonies and verify it. If we do this way, it will be a huge amount of work. So the problem is the growth of the background vector. So I have developed a new cloning method called alpha cloning. So this method is very simple. In the precursor vector, you have ampicillin resistance. And during PCR, I make introduction of the 4 guides into 3 PCRs and also introduce a new selection. After assembly of this vector with a precursor vector, this new vector has a new selection. And then, if you put the bacteria into the new selection, nothing will grow except the desired plasmid. Then you have everything that is what you want, and it's homogeneous population of plasmids. With this method, we really can clone in high throughput. And the vector, we have a lentivirus and piggyback also with puromycin and blue fluorescent protein selection, so you can use in multiple ways. And the guides are driven by 4 independent different promoters. This allows us to have a maximum efficiency in different tissue types. And finally, with this method, we can really do high throughput plasmid cloning. With 2 people, we can make 2,000 plasmid per week, and they can be used as plasmid, as viruses, as transposons. Also, as I mentioned, that in the field, we really don't care DNA polymorphism, but it's crucial. So together with Lucas Frick, we spent 1 year. We took the very nice pooled libraries, and we calculated the guides RNA from there, and really consider DNA polymorphisms, DNA guide RNA specificity. Then we take the 4 best combination guides together. And also, then we finally make these 2 libraries, in total 42,000 plasmids for gene activation and gene knockout. Because in some scenario, we don't need to make genome-wide screening. If you know the candidate is GPCR, you can directly do a subchapter of GPCR screens. So this library can be very versatile in many scenarios. Here, since the paper is already published, I want to summarize the validation of the libraries. So because we study prion disease, and the substrate of the prion protein is the cellular protein PRPC. Although previously the lab has performed 2 genome-wide screens with siRNA, with microRNA, we couldn't find which transcription factor regulates the PRPC expression. Using our libraries, we activated one by one, then measure PRPC with a pair of antibody using FRET assay. You can see that we identify 11 genes, proteins that really increase or decrease PRPC expression. In another scenario, if you have array library, you can always pull them down. And with 4 guides, efficiency should be higher. And indeed, Novartis is very interested in our library, and we collaborate with them for autophagy phenotype screening. You can see here, with comparison with Brunello, which was developed by MIT, our library identified twice the amount of the core autophagy genes than their libraries. And also, if you read our paper, we identify new autophagy modifiers that couldn't be identified by the old libraries. So this is a genome scale confirmation that our libraries, even in pooled format, is even better than the existing resource. Also, as when we were developing the library, there was a paper coming out from MIT. They developed a method called targeted epigenetic silencing. So CRISPR, when you fuse DNA message transferase, the promoter region guided by guide RNA can be methylated. Then this effect can be sustained even for 13 months, according to their research. So that is a durable effect. If you look at the CRISPR of guide RNA targeting window, it's very interesting. It's very broad. And the activation library we made, the guide is here. This indicates we can use the same guide. If we switch to CRISPR of component, we can epigenetically silence a gene. We tested with one gene called CD151. It's a cell surface protein. It really efficiently silences this gene, and also we confirm at the genome scale with a screening. Our library can also be used as an arrayed CRISPR-off library, because nobody has an arrayed CRISPR-off library. In some scenario, if a gene knockout is sensitive for the cell types, you can do genetic silencing without cutting the genome. So also, one last small thing is I want to mention. In the field, even in other fields, not always you can easily transfect cells with plasmids. So we need to make lentiviruses to enhance the delivery efficiency. So in the last 2-3 years, I also developed a high throughput method. We can make the lentiviruses in high throughput and also in considerable high titer. So the last obstacle for CRISPR screen is lentiviral template switching. So here we give us a brief summary. If you have because the bulk library, they have a mixed construct together. So the lentivirus have 2 copies of RNA genome. So when doing the packaging, if you have a mix of the vectors, you have a mix of the genomes. So it is called so-called lentivirus template switching. And before our libraries, nobody knows. We know the phenotype exists, but nobody knows quantitatively how this affects the screening. So we really tested by using the pre-pooled vector, and also we also compare with the virus made one by one and pulling together. We call it a post-pool. You can see that if with post-pooling, it really increases the sensitivity of the screening, especially for the genes has a moderate and a smaller effect. But this way of post-pooling is very laborious, and also it's cost a lot of money. Then I find a way, if you do long-read sequencing and purify post-hocly by bioinformatics the switched reads, that means you discard the cells that have switched reads, then the rest of cells, you will see, according to our results, it will see a similar results as a post-pooling without template switching. That means I also find a new way where we cost efficiently can do pool screen with much higher sensitivity. So here I want to summarize that in the last years, I really think that I solved 2 issues. First, the guide RNA efficiency is is unstable if you do single guide. Now with quadruple guide, it's really good. And the most key for the success of the library is the molecular cloning method. And our library also considers DNA polymorphisms among all the, at least 70,000 individuals. And as I have shown that our library is very flexible. You can use individual genes, array screens, put screens, and they are very efficient. And elimination of the lentiviral template switching issue really improves the performance of the CRISPR put screens. So now we really lay a foundation that we can use our libraries for biological discovery. So now I give one slide summary of what we are doing, and we have almost finished already one project for the Parkinson and Prion disease studies using the libraries. So the first question for Prion disease we addressed is because aggregates exist in the brain. How it imposes the toxicity to the cells, it is not clearly understood. Then we did a genome screen together with Tintin Liu. We did a genomic screen and find, very surprisingly, primal cilia on the neuron is super important for the toxicity of the aggregates. If you stop the cilia genesis, the slice treated with prions and also the mice treated with prions has prolonged lifespan. And also there's another question we want to address is, as I mentioned already, if the aggregates, how it spread from one cell to another cell, we call it transmission. So with Vangelis, he's a student I co-supervised with Adriano. He developed a co-culture system. You have a cells that is already being infected by prions. Then we give naive cells, and the naive cells is edited. Very surprisingly, we find a new complex called blocks is very important for the transmission of the prions. And for Parkinson's disease, this is my main focus. So with Götz-Preisträger Yin, she was a student I co-supervised with Adriano. So for the Parkinson field, the single gene risk factor for Parkinson is GBA1, which is a lipid enzyme called beta-glucocerebrosidase. So its expression goes down, or the mutation causes low activity, cause the risk increase. So the open question is, what are the transcription factors control the GBA1 expression? And what is the mechanism control the cell death? So for the first study, it was also a proof of concept. We screened all the transcription factors and identify 11 that really regulate GBA expression. And for the next second question, now I'm doing it, and I performed the genome-wide screening. I found that this GBA mutation, the core problem is a trafficking problem. It has a misfolding problem in ER, then it will not be able to traffic to lysosome. So here I really identify a core gene that can really push GBA mutant to lysosome. The new pathway has been identified. So I hope it is clear. So the last, I would say, the library is generated for neurodegeneration. However, it can be used in many ways in other biomedical research fields. So inside of the lab, outside the lab, we have a lot of collaborators. And luckily, we were supported by the Swiss Open Science grants for the share of our libraries. And this was the first retreat we had last year. And also, lastly, I want to thank, it's a huge effort because 42,000 plasmids. So the library generation involves many people. I was the leader of this, and also I supervise most of the parts. And also for the other projects, we have a lot of supports from inside and outside. Lastly, but most importantly, I really appreciate and my work has been recognized by the University of Zurich Foundation, and I'm happy to also receive other funds from, I also get a career development award from the Dementia Research Foundation, also some other funding support from Michael J. Fox Foundation, also University of Zurich. Thanks a lot for your attention. Yeah.